the chat, not to ask your question in the chat, but request to ask your question, and you will be invited to ask your question and contribute. So thank you very much. And now I will stop here and introduce the first session. Samuel Blackman, who is the founder and chief medical officer of Day One Biopharmaceuticals and Node Therapeutics, accepted the challenge and the mission to chair the first session. So Sam, good morning. Are you with us?
I am, Gilles. Good morning.
Good morning, Sam, and thank you very much. I give you the floor.
I thank you, Gilles. So good morning, everybody. It is really a privilege to be able to chair the first session and help kick off the meeting. I personally miss seeing all of you at the Steigenberger Hotel in Brussels. And I do hope that next year we will all be together in person to continue the important work of this organization. Today's first session is entitled Access to Medicines Outside of Clinical Trials in the Pediatric Precision Oncology Landscape and Capturing Data. And this is really a fascinating topic, and I applaud the Accelerate Program Committee for putting it on the agenda. From just a little bit of explanation, as you're all, I'm sure, aware, there are far more cancer therapeutics approved for use in adult oncology than there are in pediatric oncology, and as a result, it is possible, once a drug is approved, to use that drug outside of clinical trials and outside of the label. This could be off-label use or compassionate use on a named patient basis, or in the United States under right to try legislation. This, coupled with the easy availability of next-gen sequencing of pediatric tumors, has put us in a position, really one of unintended consequences, where we can potentially match children to drugs outside of the clinical trial setting. The pediatric oncology community, of course, being resourceful, is devoting efforts to figure out how to handle these unintended consequences, how to make use of this data, and how to improve the outcomes for patients. And I think that the talks that you're going to hear today will provide a good basis for the subsequent discussion. We've got four terrific speakers: Julia Glade-Bender, who is a pediatric oncologist and vice chair for clinical research at Memorial Sloan Kettering; Michael Vranken, who you've just met, who's a pharmacist by training and a member of the Accelerate staff; Jim Whitlock, who is the division head of hematology oncology at the Hospital for Sick Children in Toronto and the current chair of the C17 consortium; and Pablo Berlanga, who is a pediatric oncologist with the Institut Gustave Roussy and the head of the SASHA project. And I invite Dr. Glade-Bender to be our first speaker this morning. Good morning, Julia. You're on mute, and once you're
Good morning.
Well, I will put myself on mute. And again, please, if you have questions or want to have questions during the discussion, please raise your hand at that time. We won't take questions during the presentations. And Julia, the floor is all yours.
Good morning. Well, this is a very deep honor to be able to kick off this very important meeting. And I've been asked to set the stage of really the status right now of international efforts for precision oncology and how we go about getting drugs to children. Next slide, please. So I think most people know that many centers, many platforms now have a multidisciplinary clinical sequencing program, and the workflow works very much like this all around the world. Oncologists, pediatric oncologists are asked to educate and consent their patients, at which point a sample is acquired and processed by surgery and pathology. And then it goes through an analytic pipeline that may look very different from one platform to another, but involves pathologists, bioinformaticians, and computational biologists, at which point that information is fed oftentimes to a molecular tumor board, which has a multidisciplinary representation, and decide what results should be returned both to the pediatric oncologist, geneticist, and the patient themselves, and then decide whether or not that should direct clinical decision-making. Next slide, please. And what began as a very simple platform of next generation DNA sequencing has really become much more comprehensive, a multi-omic, if you will, multi-layered molecular tumor profiling platform, which can involve DNA from the cancer, either using a next-gen panel or a whole exome or whole genome panel, the transcriptome via RNA-seq, the methylome and methylation assays, and also many now also require a sample of normal tissue so that DNA can be extracted from the normal tissue. And from this information, one can derive what are the cancer variants, what are the germline variants, what are the cancer-specific mutations. We also get information about copy number variation and translocations, gene expression of either variants or really in comparison to reference values, as well as new tumor classifiers using methylation information. Next slide. And when we really first started doing this, the pilot studies, most of which were single institution studies and a few small consortium, what was remarkable was, first of all, that this approach was feasible, that you could get real-time information and see potentially actionable findings in really about a third to a half of patients. And then consider using these for decision-making. However, what was imminently clear from the very start was how very few patients were actually getting their matched targeted therapy. Really, with rare exception, the number of patients that were getting to therapies were in the single digits. Next slide, please. And why was that? Well, first of all, how do patients get to drugs? Well, I think we all believe that clinical trials would be the best and first choice. But when we began this, I would argue that we didn't have the right clinical trials built as yet. We were really trying to put patients on early phase trials that might or might not have had enrichment strategies. There were beginning to be biomarker cohorts within consortium trials and industry trials, but we had really not yet built what we would now refer to as basket trials, which were trials that were tissue agnostic and put patients with certain genomic alterations into baskets of drugs designed to target those alterations, which are now well known in the pediatric match trial in the US or the eSMART in Europe. In addition, we really can't do much of what the adults do, which is to have an umbrella trial, which is a histology specific trial where, if you will, different spokes of the umbrella would stratify patients with a single diagnosis like neuroblastoma into molecular cohorts. So absent a trial, how do you get a drug to a patient? Well, there are a number of non-study options, which of course end up being, if you will, an N of 1 study. You can use a commercially available agent off-label. You can do what is called a single patient use investigational new drug application, or there are compassionate use protocols, which are really, if you will, a bunch of single patient use bunched together requests, because there's very minimal data. It's just a mechanism to get the drug to the patient with no research question. Next slide. And at this juncture, I think that this schema that was published by Jones et al. Recently in Nature Reviews Cancer, I think gives the best overview of national and international precision medicine programs for pediatric oncology right now. And many countries or countries together have developed a platform which, as you can see, can have a number of molecular profiling techniques. In the US, the pediatric match went with a NGS panel and had very clear rules about what trial a patient could go on, making the molecular tumor board not necessary. This was all computer generated, whereas other countries had a much more rich set of assays and really relied on a molecular tumor board to send patients in the direction of a trial that was then available to them either through eSMART or Inform. Next slide, please. And why are the trials so important? I always draw attention back to this very first basket trial run by David Hyman, looking at tumors that had V600E mutations, but were not melanoma. And I think most people would have made the assumption really that if you had the exact same mutation of interest, of course the tumor should respond to that. And what was really remarkable was that some diseases responded very well, like non-small cell lung cancer and Langerhans cell histiocytosis, and other histologies responded very poorly, like the colorectal cancers. And it's only after really studying these tumors that we understand that, for example, colorectal cancer has an intrinsic resistance because under the pressure of the vemurafenib, there is feedback activation of the EGFR pathway. Next slide. So if you're not in the context of a clinical trial, well, what does that look like? Well, what if you used a commercial agent? I just wanted to tell you a little vignette that happened very early in my experience with precision medicine. We had a 12-year-old child with a really aggressive inflammatory myofibroblastic tumor that was ALK negative. So we submitted her tumor to the pipeline and found a novel VCAN IL-23 receptor fusion. And this predicted fusion protein was expected to have an extracellular domain of Versacan and then some extracellular transmembrane, but mostly the intracellular signaling domain of the IL-23 receptor. And so this was predicted to activate intracellular signaling by the JAK2 pathway. So of course we thought, well, why not use ruxolitinib? Next slide. And we knew the pediatric dose of ruxolitinib. Mignon Loh et al. Had published that before, and it was a 50 milligram per meter squared dose twice daily that we knew would effectively shut down signal pathway. But the FDA approved dose for adults with myelofibrosis was 20 milligrams twice daily with a maximum dose of 25 milligrams twice daily. Well, for this patient who had a BSA of 1.5 meters squared, the calculated dose that we thought would have a pharmacodynamic activity was 75 milligrams twice daily. But we could only get 25 milligrams twice daily from the insurance company, and that's what was given. So you didn't read about this working in a case report because it didn't. And the problem is that we don't know why. We don't know if the tumor was actually driven by that translocation. We don't know if at that dose the drug could effectively suppress signaling. We don't even know what plasma levels were achieved. And this is really part of the problem with giving drugs that are commercially available, is that we really don't learn much right now. Next slide, please. I'd say we do a little bit better with compassionate use and expanded access SPUs, because here we're allowed to collect some data that's clinical. And in fact, my institution will allow us to collect PK data because we call that safety data, because if we're giving the drug, we want to make sure that we're giving it in a range that is both non-toxic and capable of shutting down the pathway of interest. The problem with compassionate use expanded access drugs right now is, first of all, you need a willing partner. That is to say, a pharmaceutical company that's willing to provide you both the agent and dosing recommendations. Then it's quite labor intensive. You're really being asked to write a protocol for one, and you need to submit this to your national regulatory body. And in the case of the US, that's the FDA, as well as your institutional IRB or ethics board. And this is a completely unfunded mandate. So once you do all of this work, and I had my research staff keep track, and it was about 40 person hours of work, the institution would need to provide the research pharmacy resources, as well as clinical oversight, research compliance, and you have to report the adverse events here as if it's on a phase 1 trial. And still, there's a lost opportunity for data collection because it's really not a research study, it's a treatment plan. And so that it's only that minimal data that's captured, and it's not clear where that minimal data goes after it's captured. Next slide, please. But here I would say, at least with the compassionate use, we can see signals of efficacy because we know we're giving the drug at the right dose. And here is an example of a patient that we saw with what was thought to be congenital mesoblastic nephroma, which turned out to be a quite aggressive fibrosarcoma, and our precision medicine impact testing at MSK showed that this patient had a unique SPEC-RET driven tumor and had failed really chemotherapy. It was quite toxic and the tumor was not responding. We made a single patient IND application to receive selpercatinib before the pediatric trials were open, and it was FDA approved, and really saw quite a remarkable response, including tumors that were in the CNS, which was very important. But over time, these recurred, and we were allowed to escalate the dose, and they subsequently re-responded. So we learned a lot from this single patient. Next slide, please. And actually, there were 5 patients during this time that Loxo allowed for single patient INDs, and my colleague Mike Ortiz, along with the Loxo group, were able to publish this preliminary data as a case series. There were 5 patients who received this, all 5 had responded, and there was some good PK data that demonstrated that kids were achieving doses in the same range as the adult recommended phase 2 dose. And this preliminary data was really good basis for developing what are now pediatric trials of this drug that are available through the MATCH platform and others. Next slide. So now, in terms of the national profiling programs, we're beginning to see really large numbers of patients with molecular profiling. And so, and there are a lot of patients who have been matched to trials. I would still say the minority are receiving matched targeted therapy. It's more like a third, and you can see here that trials that are directly linked to basket studies are actually doing a little bit better, but it's still no more than a third of patients that are reaching their matched targeted therapies. So I think there is room to do a lot better. And when we do so, next slide, I think we really need to address many of the gaps now that are in the international platform. And that is that we do not have a programmatic, centralized international repository for the genomic data of these patients and the targeted agent experience, including these real world data situations. Nor do we have a place to catalog in a programmatic way functional and clinical validation of what we presume are targets, and also do not know these variants of uncertain significance. We also need more granular data on our matched therapy. What was the agent that we used? What was the means of access? What was the dose, the toxicity, the response? And I feel very strongly this must include negative data, because if each of us are doing independent, compassionate use or commercial use agents throughout the world, and only the one responder gets published, then we're really not seeing the full results of this real world data. And more children will be exposed to mostly ineffective agents. And finally, we really need the ability for our industry partners to be able to leverage this real world data in the development of their pediatric implementation plans and their initial pediatric plans. And that's where I wanted to leave things for now, hoping that we'll have a lot of time for some interesting discussions. So thank you very much.
Julia, that was absolutely terrific. Thank you. That latter point on, the importance of disseminating negative data is, I think, so critical, and I'm so glad that you raised it. And we'll talk about this more in the discussion. And with that, I'm going to invite Michael Franken to join us, who's going to talk about work that he's done defining an international legal framework for compassionate and off-label use. Michael, thank you for joining us this morning, and thank you, congratulations on joining the Accelerate team.
Thank you very much. It's an honor being here and working for all of you. Next slide, please. So regarding experimental medicines and off-label use, how do patients gain access to innovative medicines outside of clinical trials? Well, there are mainly two pathways, the ones of the experimental drugs on one side and the approved medicines on the other side. Next slide, please. First, we're going to dig deeper into the experimental drugs, which are accessible through first the compassionate use program, or as it also is called in the USA, the expanded access program, in which a medicine which is not fully evaluated in clinical trials can be made available for an unmet medical need. However, safety and efficacy profile needs to be required. Then we jump to the named patient base in the EU. This is for a single patient, also for an unmet medical need in which doctors gain the medicine directly from the manufacturer. And in the USA, there's also the Right to Try Act, also for access to experimental drugs, in which it's possible to exclude the FDA from request process, and also here the FDA did not yet determine the full safety and effectiveness of the experimental medicine. Next slide, please. Regarding the approved medicines, there's off-label use. Off-label use of medicines is the use of medicines outside the terms of their marketing authorization or their dossier, which is prescribing a drug different than its original indication or a different dosage, for example. Next slide, please. Regarding compassionate use around the world, in Australia, there's the special access scheme in which they evaluate case-by-case patients. There are three pathways to address this unmet medical need. In Canada, there's the special access program in which it's noteworthy to say that there's the future use request for future medical emergencies. In USA, I already explained. So next slide, please. In Europe, the European Commission granted the regulation for compassionate use some 10 years ago. So they tried to facilitate and improve access to this type of programs, and the national member states adopted this. Then I'm going to look further into the member states, for example, Germany. In Germany, there's the compassionate use program where it's noted to say that the National Competent Authority, BfArM, is responsible and takes advices from the Paul Ehrlich Institute. In France, it's noteworthy to say that there's a note in the wording, which is efficacy and safety should be strongly presumed instead of established. In Lithuania, for example, there's no compassionate use available, or at least I didn't find it. As well as in Malta and in Cyprus. So there are some countries in Europe which do not yet have a compassionate use program. But for most of the high income countries, there is a legal framework for compassionate use. But there's also some heterogeneity in this framework. Next slide, please. There was a paper published in which they surveyed 132 pediatric oncologists in the United States. And I identified some common barriers towards this early access program. The ones they reported were inability to identify a drug that had the potential to help patients, lack of knowledge, experience, or understanding of this application process, and a high burden of administrative workload in this application process. Next slide, please. However, there's also some help towards patients and healthcare professionals in terms of facilitation programs for compassionate use, such as FDA Project Facilitate aimed for oncologists, and the Kids v Cancer Compassionate Use Navigator for the pediatric oncology community specifically. Next slide, please. Jumping back to off-label medicines. So prescribing off-label is very frequent in pediatric population. And whenever I read a paper on off-label use, this was highlighted. In Europe and in the US, this is not regulated, but common practice, and is often a responsibility on behalf of the prescribing physician, laying sometimes a heavy burden on them. There's also some reimbursement issues because reimbursement and pricing is often linked to the dossier of the medicine. Then there's also higher rates of adverse drug reaction due to off-label prescribing. Next slide, please. To conclude, what I all said is that a legal framework for these access to experimental medicines exists, but is different and complex around the world. There's some heterogeneity in Europe regarding the regulatory instruments in the access to these experimental medicines. So the questions within Accelerate is what we wonder to ourselves is how this data is captured on safety and efficacy for both compassionate and off-label uses, and how this valuable real-world data could potentially be available to suggest further evaluation of a drug. So the first thing we're going to do together with ITCC is survey healthcare professionals to determine current processes and data capturing in Europe, Canada, and Australia. And with this, I thank you for your time and your attention.
Great, thank you very much, Michael. Next up, we have Jim Whitlock. Jim, you're with us this morning.
Yes, good morning, Sam.
Hi,
Thanks for the
Jim.
opportunity to talk about the Canadian experience with what we call single patient protocols or single patient studies. So I'm presenting on behalf of my colleagues in C17. Next slide. Next slide. So in Canada, access to novel therapies for children with cancer outside clinical trials is a real challenge, particularly given the fact that we're not able to participate in the many available clinical trials elsewhere in North America, such as the MATCH study and a number of early phase studies through the Children's Oncology Group. Julia and Michael have nicely outlined the pathways that are available, so I won't belabor that point. If a commercial drug is not available, we do have a special access program, which is essentially permission to import a drug that's not approved in Canada. Funding and other issues can often preclude that. This pathway does not work for all drugs. For a drug that has no approval anywhere, or if there are inadequate data, or increasingly, if it's we are proposing to use a drug as part of a novel combination, then the SAP program doesn't work, and we run into obstacles. So I'm going to share with you what I think Julia referred to as an expanded access mechanism, which we have been working with our regulatory colleagues at Health Canada to address, called the single patient study. So it is a clinical trial. It requires a clinical trial application to Health Canada. It does require an ethics view, and it has to have a sponsor. Here, C17, the national organization, has served as a sponsor of a growing number of these studies. Next slide, please. So single patient studies under the regulations that we operate under are intended for patients with serious life-threatening conditions with no other treatment options, no other way to access the proposed treatment. It is a clinical trial, and it so has to be compliant with national regulatory requirements. It only allows a single patient. We can submit an additional protocol request for another patient, which would use exactly the same therapies, but it has to be a separate application. It requires pharma partnership for drug access and IB. We've been having pre-submission teleconferences with Health Canada to try and smooth out this new process, which has really worked well. Again, the PI writes a protocol and consents and is responsible for submitting safety reporting with no support, as Julia mentioned. The sponsor here, C17, prepares the clinical trial application, assures monitoring, and then the institution has a number of responsibilities for various levels of review. Next slide, please. So if you could click through 5 clicks on this animated slide. This is the first example of a trial of palbociclib and temozolomide and irinotecan, which ironically is available as a clinical trial in the Children's Oncology Group, but we did not have access to that. So a patient who was diagnosed in September 2018 was enrolled subsequently in our National Profiling Program profile, had a molecular tumor board identifying potential targets in January 2019, and then relapsed in January 2020. On that day, C17 was contacted about assistance in submitting a single patient protocol. Pharma and Health Canada were contacted the next day. Pharma approved to supply the investigational drug within a period of 2 weeks. Health Canada, then the protocol was prepared and submitted to Health Canada on the 11th. They approved it within 48 hours with a no objection letter. It was submitted to the ethics board and approved, and this patient was able to start therapy on March 2nd, approximately 6 weeks following the time of relapse, which in some situations would be a very long time. And in this period of time, it was remarkably quick. Next slide. So we've now submitted a total of 5 centralized single patient studies. You see the drugs listed here. It's been working for us. And some of the advantages of this approach is that we can now get timely access to selected novel agents for some of our patients. I think our pharma partners here in Canada have found it valuable because they have assurance of monitoring, safety reporting, and because it's a protocol in a study, they have access to the data, which compassionate use does not really address. And as Julia very nicely pointed out, this approach can identify promising clinical signals for further investigation, perhaps, exceptional responders, et cetera. But there are many obstacles to this new approach. As you've heard, the workload is significant by definition because it's restricted to a single patient. It's not really scalable. Our efforts at centralizing this at a national level and working to standardize the process have, I think, reduced but not eliminated some of these obstacles. And again, there's a question of what this research really contributes to the field in the absence of a way to systematically share the data. And so I want to fully support the proposal that Julia made for a global registry of such studies so that we can share data and avoid redundancy. And finally, here in Canada, the SPS approach works, but it has not solved our larger drug access issues, which we continue to address. So next slide. I'll stop there and thank all of my colleagues who've done all the real work in this, Daniel, Rebecca, and Ty, who've written a number of these protocols, Leah and Kathy at the C17 office who've overseen this process, our colleagues in Health Canada who've worked very closely with us to put this into place, and finally, our pharma industry partners in Canada who have supported these requests today. Thank you.
Jim, thank you very much for going through that, and really kudos to the team in Canada and C17 for organizing this and really creating an innovative infrastructure. Our final speaker today for brief talks is Dr. Pablo Berlanga from the Institute Gustave Roussy, who's going to talk to us about the SASHA project in his institution.
So, hi, good afternoon to all of you, or good morning or evening, depending on where you are. So first of all, thank you very much for letting me present the SASHA trial of the study of the France. So first of all, just a quick overview of the current access for children and adolescents with cancer to therapeutic innovation in France right now. So it's more or less follows what Julia said before. So in case of disease relapse or progression, there is a discussion and proposal of tumor molecular profile. The MAPIAX trial has already closed, and we have now the opportunity to do that within the France Médecine Génomique 2025, which is considered clinical care and supported by the Health National System. But there is also a research trial that is the continuation of the MAPIAX to reply to some of the questions, like, for example, liquid biopsies. Then results are discussed in a national molecular tumor board. The idea is to give adequate therapeutic recommendations based on the molecular profile. It's important to say that there is a ULink, that it's a portal that has been developed by parents associations with the help of the French Society of Pediatric Oncology, in which parents and physicians can find the updated information of clinical trials, biomarker-based or not. Because at the end, the idea is, of course, to try to include patients in trials to generate the best information. As Julia also said, that there is this biomarker-driven basket trials as the ACCESS SMART that has been developed 4 years ago, and that covers target therapies that cover most of the pathways that we find in this molecular profile, tumor molecular profiles performed at relapse. But up until now, at least in France, we had no way to know what happens with those patients that we have said that may receive an innovative cancer therapy outside marketing authorization and outside clinical trials. So this is why we have developed in France this SASHA study that has been developed by the New Drug Development Committee of the French Society of Pediatric Oncology in collaboration with the current structure in France of the cancer care of patients, which is the Interregional Pediatric Oncology Networks. So this study, it's a French observational study that collects prospective data of toxicity and efficacy, data of off-label and compassionate administration of anti-cancer therapies that are administered to children, adolescents, or young adults less than 25 years of old with pediatric tumors outside the frame of clinical trials. Of course, the idea is always to put patients in trials whenever feasible. So one of the things that we have developed within SASHA is that patients need to be discussed. We reinforce the discussion of patients in the interregional multidisciplinary tumor boards to ensure that there is not a clinical trial in which the patient could be eligible or included, and then in case it's not feasible, then support the proposal to give the off-label or compassionate use drug. That is always the decision of the referent physician. And we collect the data of toxicity and efficacy in a validated pharmacovigilance tool that has already been in use in France for several years, that is called Viginome. So here, just briefly, in case of patient disease relapse progression, it's very important that the patient report is discussed in a multidisciplinary interregional tumor board to ensure, first of all, if feasible, clinical trial inclusion, and if not, off-label compassionate use may be recommendation, and try to collect this data in the SASHA study. So SASHA is a reality that opened in March last year, and so far we have included more than 100 patients. So for us, it's important because it covers an important need. And here, this is the previous slide, and now with SASHA trying to cover that small part. So just to finish, thank you very much to all the people in the French Society of Pediatric Oncology, in the Scientific Committee, SASHA, Pharmacovigilance Unit, and Gustave Roussy, New Drug Development Committee, and Precision Medicine Committee, and of course, the funders that have allowed to the SASHA study to start, Imagine for Margot and Fondant Cancer, and Fondation Dulem, that it's a federation of French pharmaceutical companies that have also supported this SASHA study. So thank you very much.